Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 1 for May 2020. This is the Part 1 video. I'll put the link to the Part 2 video below in the description box. Let's begin with question 1. Question 1 says, the primary structure of a protein determines its secondary structure and its three-dimensional structure. Read through the following account of the primary structure of a protein. They say complete the account by writing the most appropriate word or words on the dotted lines. The primary structure of a protein is the specific sequence of amino acids joined together by, of course, these are peptide bonds. These bonds are formed between the amine group of one amino acid and the carboxyl group of an adjacent amino acid by condensation reactions. Actually, you could switch that and that around. This could be carboxyl and this could be amine. These bonds are formed during the stage of protein synthesis called translation. Translation is the process when the amino acids are linked, water is lost, and then a peptide bond is formed in between the two amino acids. In Part B, they say, the table describes the type of bond that holds the secondary and the three-dimensional structures together. Which type of bonding is true for each structure? The secondary structure is, of course, held together by hydrogen bonds only. And then the three-dimensional structure is held together by both hydrogen bonds as well as ionic bonds. So this brings us to the end of question 1. Let's continue to question 2. Question 2. Most Bengal tigers are orange with black stripes, but there is a very small number of Bengal tigers that are white with black stripes. The photograph shows a white Bengal tiger with black stripes, which is this one here. White tiger offsprings are produced by two Bengal tigers that each carry at least one recessive allele for a gene affecting coat color. The pedigree diagram shows the phenotypes of one family of tigers bred in captivity. This was given to us as a pedigree diagram. This is a female orange tiger and that is a male orange tiger. So we can see both of these are orange, however they have children or offsprings that are white. So that means they are both heterozygous because they can produce offsprings that are white. So we can decode the genotype of the parents. We can also tell that this is orange, 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 and this is going to be white. Next they say the phenotype is affected by the genotype. State what is meant by the term genotype. Genotype is the combination of alleles. Moving on. Here they say, state the probability that the next tiger born to these two parents will be female. Of course, the probability of having a female is a half. In the next part, they say, determine the expected phenotypic ratio of orange tigers to white tigers born to parents shown in this pedigree diagram. Use a genetic diagram to support your answer. You need to remember that from the pedigree diagram, both parents were heterozygous, and if I like their genotype to be that and that, it means the possible gametes are going to be that, as well as this and that, and the possible genotype of the offsprings is going to be that, and that. So down here I said expected genotype is going to be that, and that. And then the phenotypic ratio of offsprings is going to be 3 orange and 1 white. Here they say the incidence of white tigers in the wild is 1 in 10,000 Bengal tigers. There are approximately 6,000 Bengal tigers in captivity, 200 of which are white. They want you to calculate the incidence of white tigers in captivity. Since we have 206,000, the ratio is going to be 1 in 30. This brings us to the end of question 2. Let's continue to question 3. Question 3. The cardiac cycle is a sequence of events that occurs when the heart beats. A typical cardiac cycle takes 0.86 seconds. The diagram illustrates the cardiac cycle, which is this one here. And for the key, we have atriosystole, atriodiastole, ventricular systole, and ventricular diastole. So here they say which row of the table describes the atria and ventricles during atriosystole. Atriosystole is the contraction of the atria, so we expect the atria to be contracted and the ventricles to be relaxed, so B should be the answer. Next they say, explain why there is a delay of 0.01 seconds between atriosystole and ventricular systole. That delay is there to allow the valves to close. So I say to allow the atrioventricular valves to close before the ventricles begin to contract. And the closure of these valves prevents the backflow of blood from the ventricles into the atria. Moving on. Here they say, using the information in the diagram, calculate the duration of ventricular systole in milliseconds and express your answer in standard form. So if we go back here, we can see ventricular systole takes place in 3 out of 8, meaning we're going to have 3/8 times the duration, which is 0.86, and the answer is going to be that in seconds. However, we know that 1 second is that milliseconds. Remember, our answer has to be in milliseconds, so 1 second is 1,000 milliseconds, and therefore 0.3225 seconds is going to be that times 1,000, which is that milliseconds. To write this in standard form, so it's going to be 3.225 times 10 power 2, which is rounded off to 3.2 times 10 power 2 milliseconds. Next they say, state what proportion of the cardiac cycle is spent in ventricular diastole. Again, if I take you back here, ventricular diastole is going to be this, which is 5/8. So 5/8 times 100 gives us 62.5%. That is the percentage. And down here they say a typical cardiac cycle takes 0.86 seconds. During exercise, the heart rate increases and the duration of cardiac cycle decreases. Calculate the increase in heart rate if the cardiac cycle decreases by 0.4 seconds. So here I calculated first for when the heart rate lasts 0.86 seconds. I said in 0.86 seconds we have 1 beat, which is 1 cycle. And therefore in 1 second, which is 1/60 minutes, we will have that divided by 0.86. And in 1 minute, we will have that times 60, which gives us that. That is going to be the heart rate. Now, if there is a decrease by 0.4, it means the new rate is going to be at 0.86 minus 0.4, which is 0.46. And therefore, the rate will be 60 divided by 0.46. This is similar to what I did here, because this was also 60 divided by 0.86. So 60 divided by 0.46 gives me this. That is the new heart rate. Therefore, the increase is going to be that minus that, which gives me 61 as the rounded off answer. So this brings us to the end of question 3. Let's continue to question 4. Question 4. The polynucleotide DNA is composed of mononucleotides linked together. 2 polynucleotides form a DNA molecule. The diagram shows part of a DNA molecule, which is this one here. So first they say, draw a circle around one mononucleotide that includes the base labeled R. If this is the base labeled R, this is going to be the mononucleotide in which R is, because you can see that is one mononucleotide. That is going to be the next, and this is number 3. So circling this will be the correct answer. Moving on. Here they say, which row of the table identifies the bonds between S, T, and U? Going back here, S is a phosphodiester bond, this is a covalent bond, and that is a hydrogen bond. So phosphodiester bond, covalent and hydrogen, the answer here should be a D. Next they say the base labeled P is adenine, which is the base labeled Q. Again, to take you back, if this is adenine, that is going to be thymine, because this is DNA, so the answer should be C. Moving on. Here they say, Matheson and Stoll carried out experiments that provided evidence for the semi-conservative replication of DNA. Heavy nitrogen, which is nitrogen 15, and light nitrogen, which is nitrogen 14, were used in these experiments. The flowchart summarizes part of one experiment performed by Matheson and Stoll. So in stage 1, we have bacteria grown for several generations in a culture of medium containing the heavy nitrogen, and then bacteria from stage 1 grown for 1 generation in the culture medium containing nitrogen 14. In stage 3, bacteria from stage 2 grown for 1 generation in a culture medium containing the light nitrogen, and in stage 4, bacteria from stage 3 grown for 1 generation in a culture media containing the light nitrogen. After each stage, a sample of DNA was taken from the bacteria and the DNA molecules separated using a density gradient in a tube. The heavier DNA molecules form bands lower down the gradient than the lighter DNA molecules. The height of each band is proportional to the percentage of DNA molecules in the sample. They want you to complete the diagram to show the results of this experiment and use the dotted lines to help you to position the bands on the diagram. The first one has been done for you. Now here I have a summary of mine. In stage 1, you can see everything was cultured in nitrogen 15. So we see it's going to be heavier down the line. And then in stage 2, they all carried out experiments just in nitrogen 14. So since this is the semi-conservative model, it means after each strand splits, there is going to be a combination of nitrogen 14 and 15. So they're going to be halfway through. In the next stage, we can see there is going to be that splitting into that and that into that, that into that and that into that. And when they all combine with nitrogen 14, which is that, and that, we can see the ratio is going to be 2 pure nitrogen 14s and 2 mixtures. That is why you see there is the lighter one, which is nitrogen 14, and the mixture here. So the concentration or the thickness is going to be the same, half in half. In stage 4, I'm going to split everything here. So that becomes that, that becomes that, that becomes that, that becomes that, that, and that. And then they each pair with a nitrogen 14, which is a lighter nitrogen. We can see the ratio is going to be 1, 2, 3, 4, 5, 6. Here we have 6 that are pure, and we have only 2 that are hybrids or that are a combination of the two. So the ratio is about that to that. And that should be how you represent your data. Moving on. Here they say the experiments of Mesoson and Stowe disproved the dispersive theory of DNA replication. The diagram shows the expected results if the dispersive theory was correct. In stage 1, one DNA molecule containing the heavy nitrogen, which is that, and the appearance of the bands on density gradients will be that. In stage 2, 2 DNA molecules containing heavy nitrogen, 14, and the light nitrogen, 15. The question here says, which diagram would show the bands of DNA molecules of the density gradient at stage 2 if the dispersive theory was correct? Here the DNA molecules will be either light or they'll be heavy, and therefore the corresponding answer should be a B. So this brings us to the end of question 4, as well as to the end of the first part of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.